Hi, my name is Manish Gupta, and in this video, I'm going to talk about this new model called as LongVIT, which is basically a model to find image representations using long sequences for computational pathology. Okay, so this is a special kind of a model. In fact, the model is generally applicable for other use cases also, but at least in this paper, they sort of focused on computational pathology. I myself worked on computational pathology for some time, and the way this works is essentially, let's say, if you have a particular pathological sample, you first build a microscope glass slide out of it. I mean, the same kind of microscope slides that we experimented with in school days, right, in biology classes. And then the doctors actually spend about 30 to 40 minutes looking over the entire glass slide so as to figure out if this person has cancer or not, or figure out the grade of cancer and so on based on the tissue, which is smeared on that glass slide. Now, these glass slides, of course, they can be scanned. So the scans are typically very large in size. So a typical scanned whole slide image, as it is called, it could be pretty large, on average about a GB in size. That's that, right? So you take this gigapixel image, right? And typically the standard way of figuring out whether this particular image has a particular kind of cancer and what grade of cancer is to actually take the image, divide it into different parts. You can call them as tiles in that senses or patches in that senses. Then you basically pass each of those through some model because any model that you take in today's world cannot really process like a gigapixel kind of an image as input, right? So therefore you break them into tiles, maybe 1,000 cross 1,000 tiles or something of that kind, and then pass 256 cross 256 tiles as it is shown here, and then pass them through some image processing model, for example VIT, or you could basically also use your favorite ImageNet kind of models like DenseNet, ResNet, and so on. And then you can basically get a prediction per tile, right? You can get a prediction per tile. Those predictions per tiles, now these could be just predictions, binary predictions, yes, no, or they could be CLS vectors, the classification vectors from these different models. And you can aggregate on top of them so as to come up with the final decision. So final decision of whether this image contains a kind of cancer or not, and which grade and so on. Okay. Now this aggregation could basically just be averaging, or it could be attention-based aggregation and so on. So using transformers, right? Now that has been the traditional way of doing things. So the top method that you see is basically a traditional way of doing things, which has two stages. The first stage is to extract features using a vision encoder for each of those tiles separately or patches separately. And the second stage is to aggregate those features. Yeah. Now, on the other hand, what these guys actually have proposed is to use LongVIT encoder. Now, they basically say that we'll take the entire gigapixel image, we will actually split it into different tiles or parts as you see them, and then we will get these millions of patches, and then we'll pass them through a transformer encoder, the LongVIT encoder, okay? Which basically is going to essentially give you gigapixel image representations. So they're going to give you gigapixel image representations, which then you can actually just connect with the dense layer so as to predict whether it has a particular kind of cancer or not, and so on. Okay. So of course, LongVIT works because long sequence-based transform models are available, right? So that is why they are able to basically, make use of LongVIT for this pathology, for this automatic classification of pathological images, specifically, for cancer subtyping. I mean, basically, they do two different kinds of tasks that I'll talk about on the next slide as well: cancer subtyping and survival prediction. Okay. So what is LongVIT? Well, LongVIT is basically a vision transformer to process gigapixel images in an end-to-end manner, and it is supported, and it sort of works because in today's world, you can actually have transformers which can take a million sequence length as input. Okay. It can, since it can actually capture such a large sequence length, it can capture both short range as well as long range dependencies from millions of such patches. Okay. So therefore, now you have to, if you have to make use of LongVIT for these pathological images, you have to basically pre-train it in a way such that it works for pathological images. And second, then you can fine tune it on specific tasks that you have in mind. Okay. So for pre-training, they basically used 10,000 such diagnostic slides, microscopic slides, right? From the popular TCGA corpus. So TCGA is the Genome Cancer Atlas, right? And it has basically several of these whole slide images. They took 10,000 of them, And they used what is popular in today's world, dyno loss. So essentially the self-distillation with no labels kind of pre-training loss. So it's essentially pre-train pre-train the LongVIT model. Now this is a completely unsupervised thing, right? So essentially they just took 10,000 class slides or diagnostic slides from TCGA and pre-trained it. Okay. These 10,000 slides actually talk about 30 different types of cancer, and they basically took it at a 20x objective. So remember in school days when you used microscope, you basically had various levels of resolution. So they basically looked at a 20x resolution with a patch size of 32 cross 32. Yeah. So that's how pre-training was done. Now for fine-tuning, they actually fine-tune on two different kinds of tasks, cancer subtyping tasks and also on survival prediction tasks. Okay. And in fact, they have 3 data sets per task. So overall, it's like 6 different data sets that they experiment with. These are like labeled data sets. Okay. So 3 cancer subtyping data sets and 3 survival prediction data sets. I'll talk about those details about these data sets on the next slide. Okay. So they take that whole slide image and they resize it to a very large size, something like, let's say 32,768 squared, right? So height is 32,768, width is 32,768 as well. Okay. They, as mentioned in the previous slide, right, they basically divide the image into a sequence of millions of fixed size patches. They generate patch embeddings via linear projection, like you do in a VIT model typically, right? They add learnable one-dimensional position embeddings, again, like a VIT model. And long VIT is possible because of this LONGNET model, which can actually handle long sequences. Okay. So they use LONGNET to actually model long sequence to obtain patch representations that contain the contextual information, right? So essentially it has like long range as well as short range dependencies. Lastly, they average pool the patch representations to get the WSI, the whole slide image representation. So once you have like millions of those patches, of course, you get millions of those representations. You're basically coming out from the LONGNET model, the last layer of the LONGNET model, which is an encoder model, right? Transformer encoder model. And they basically then average pool the patch representations just to get the whole slide image representation. So how does this LongVIT model perform on this cancer subtyping and survival prediction tasks? So remember, these tasks are actually pretty difficult to do. In fact, they also suffer from imbalance. So a whole bunch of these data sets are imbalanced in nature, right? And the way the task is that if you basically look at a glass slide, whole slide image, and you divide it into millions of patches. Now those the markers of the cancer are available only in a few patches. Many of those other patches may just look absolutely normal. So that is how these patches, these images are, right? So the idea is that even if you find a few of those patches which are cancerous in nature, then you can actually mark the entire WSI, the entire whole slide image as abnormal, okay, or as a particular kind of cancer, okay. So this basically makes us think that this is more like a multi-instance learning kind of a problem. So if you are old school, you might remember multi-instance learning setup. So the setup is that if you are given a bag of instances, even if one of them is positive, you must mark the entire bag as positive. So that's basically the setup, multi-instance learning setup. And as you can observe, several of these baselines are multi-instance learning kind of baselines. And that's just because of the setup of the problem, that even if one tile in this entire large slide is basically positive or cancerous in nature, you must mark the entire image as cancerous in nature. Okay. So they experimented with two kinds of tasks I already talked about, cancer subtyping and survival prediction. Specifically in cancer subtyping, they looked at 3 different data sets: invasive breast carcinoma, non-small cell lung carcinoma, and renal cell carcinoma, right? So these are 2-class or 3-class problems. And what is reported here is macro averaged AUC scores. They worked with 32,768 square input images for BRC and NSCLC, which basically means this is the sequence length that they had to experiment with, 1 million plus sequence length, right? That's the number of patches that you obtain when you cut such a very large image into small pieces. And NSCLC, they took a 16,384 squared input, and they end up with a sequence length of 0.26 million sequence length. Okay. So as you notice, of course, the LongVIT method does the best because it can actually capture both small range and long range, short range and long range dependencies. And it is actually very, very substantial improvement across all of these tasks. So yeah, for NSCLC, maybe it's not too large an improvement, but if you look at BRCA, of course, the improvement is pretty huge, right? Also in RCC, the improvement is pretty huge, significant improvement, 1% point improvement. Remember, 1% is super important when it comes to medical applications, and that too when it is like 98 versus 99. Right? Survival prediction, they experimented again with 3 different kinds of data sets. You can read them about them here. Uterine corpus endometrial carcinoma, lung adenocarcinoma, and breast invasive carcinoma. Right? Carcinoma typically in medical world means cancer. Right? So that's that. And they basically, they did not use any genomic features as such, although some of the baselines actually make use of genomic features also. So besides the actual image, they also make use of genomic features. Okay. So LongVIT, in spite of not making use of genomic features, beats all the baselines, right? And the values which are shown here are concordance index. So that's the metric that they popularly use for survival prediction, which is basically what is plotted here. So for different kinds of tasks, they basically used different kinds of image sizes and patch sizes. Specifically for LUAD task, again, they used 1 million plus sequence length or patch and number of patches in that census. Okay. So that's that. In summary, in this video, I talked about LongVIT, which basically scales the vision transformers using the LONGNET model to gigapixel images in an end-to-end manner, and thereby supports this very critical pathology cancer predictions, right? So they use LONGNET, as I mentioned, to encode long sequences, thereby capturing both short and long range sequence dependencies, right? And they experimented with the 6 different data sets across 2 different pathologic classification tasks or pathological tasks, one of them being cancer subtyping classification and the other being survival prediction. Okay, so that's it for this video. Hope you liked the video. Thank you for watching. Connect with me on my LinkedIn or look at my research on my homepage. Thank you.